Welcome to section 10.1, Cloning and Genetic Engineering. In this section, we'll explore the basic tools scientists use to study and manipulate DNA. These techniques include isolating DNA, amplifying it, cutting and joining it, and even using it to create genetically engineered organisms or clones. By the end, you'll see how these methods are used in medicine, agriculture, and research. By the end of this section, you should be able to explain the basic techniques used to manipulate DNA, like PCR and gel electrophoresis, and describe the difference between molecular cloning and reproductive cloning. Biotechnology is all about using artificial methods to modify genetic material for useful purposes. When you hear this word, think about the broad range of applications it has, from developing better medicines to engineering crops that resist pests, and even industrial uses like producing biofuels. The key idea is that scientists can now move beyond simply observing DNA. They can actively manipulate it, cutting, copying, and transferring it between organisms. This ability has opened the door to genetic engineering and cloning, which we'll explore in the rest of this section. The first step in working with DNA is isolating it from cells. Scientists break open the cells, remove proteins and lipids, and then separate the DNA. The result is purified DNA that can be used in experiments. Imagine this as removing the instruction manual from a locked safe so you can actually read it. Gel electrophoresis is a technique that separates DNA fragments by size. DNA is negatively charged, so when an electric current is applied, DNA fragments move through a gel. Smaller fragments move faster and travel farther than larger ones. This method allows scientists to analyze DNA, compare samples, or check whether an experiment worked. It's like sorting beads through a sieve, with small beads moving more easily. Polymerase chain reaction, or PCR, is one of the most important techniques in biology. It makes millions of copies of a specific DNA segment. The process has 3 steps repeated in cycles: denaturation, where DNA strands separate, annealing, where primers bind, and extension. DNA polymerase builds new strands. PCR is used in forensics, medical tests, and research. For example, PCR was the key to rapidly detecting COVID-19 infections. Molecular cloning means copying a gene and inserting it into another organism. A common example is inserting a human gene into bacteria so the bacteria can make a human protein, like insulin. Scientists cut DNA with restriction enzymes, paste it into plasmids, and then let bacteria do the work of expressing the gene. This shows how we can harness living cells as protein factories. Reproductive cloning is different from molecular cloning. It creates an entire organism that is genetically identical to another. The famous example is Dolly the sheep, cloned in 1996. Scientists transferred the nucleus of an adult sheep cell into an egg cell that had its nucleus removed, then implanted it into a surrogate. Dolly proved that specialized cells could be reprogrammed to create a whole organism. Genetic engineering uses these tools to create genetically modified organisms, or GMOs. In agriculture, GMOs can be crops that resist pests or tolerate herbicides. In medicine, Genetic engineering produces human proteins like insulin or clotting factors, and even gene therapies. These applications show how cloning and genetic engineering move from the lab to the real world, affecting food, health, and society. Scientists use a set of core tools to study and modify DNA. First, DNA must be isolated. Gel electrophoresis and PCR allow scientists to analyze and copy DNA. Molecular cloning inserts genes into new hosts, often bacteria, to make useful products. Reproductive cloning produces entire organisms, as with Dolly the sheep. Finally, genetic engineering applies these techniques to create GMOs, produce medicines, and develop therapies. Together, these methods show the power of biotechnology.